In this chapter, we are going to talk about the viruses and other acellular infectious agents like the prion proteins. So, in this chapter, we will understand that what the viruses do. This is a very unique group or class of organism, and viruses actually do more than cause a disease. And in the recent times, after the COVID-19 pandemic, the virology is again all over the place. Even in the street, everybody are talking about the virology. So this is indeed a very important chapter, and we will have a continuation of this chapter into the next and the last chapter of our Unit 1. So interestingly, if you just take a look at the textbook, and there was a case that was reported that one patient in Great Britain was suffering. She was a cystic fibrosis patient, so he has a very thick mucus lining her the bronchus and the bronchioles. So it is difficult for them to breathe as such, and they suffer a lot of different type of infection. And she was chronically infected with a species of bacteria, which is Mycobacterium. Now, the Mycobacterium tuberculosis caused the tuberculosis, and this is TB. But there are several other respiratory Mycobacterium that can cause, and the one that was infecting her, or was chronically she was infected, was Mycobacterium abscessus. Mycobacterium abscessus causes an infection and, of course, cause lesion to the lungs, but it is not tuberculosis. So it is a non-TB Mycobacterium species. She had to undergo a lot of medical treatment for more than 8 years. She had to undergo the medical treatment, even needed lung transplant, but nothing. She did not, the doctors could not get rid of these Mycobacterium because it was resistant to all of the antibiotics. But finally, there was a laboratory in the Pittsburgh in Pennsylvania. It was found that they do have an isolation of bacteriophage, and these are the viruses which infect the bacteria. And particularly, this isolate in Pittsburgh was isolated from a Mycobacterium abscessus. What it means that this particular bacteriophage infects and lies or kills the Mycobacterium abscessus. Now, this is known to the human being for a long time, that the bacteriophages do kill the microorganisms, and it is widely used clinically in the Soviet Union and some of the parts of the Eastern Europe. But in the United States, there is none of these bacteriophage treatment has been approved, and it is not available yet in the United States. So what it was done, that this bacteriophage finally was actually injected to her, because it doesn't cause any harm to the human beings. These are bacterial viruses, and the Mycobacterium essentially succumbed to this bacteriophage, and she was out of this condition. And at the later part, almost a similar kind of a infection that was essentially that happened, and in the United States, and in experimental basis, there was a second story, which is a following story, where the Mycobacterium abscessus infection was cured by this bacteriophage. So there is a paper which is directly related to this, and it is uploaded into your supplementary information, so you can take a look at that. So in addition to these ideas that why the bacteria, so this is first, you are just being represented by a bacterial virus, which is beneficial to us. But along with that, of course, they cause disease, but many of them actually do not. But all of the plants, fungi, bacteria, even archaea, of course, the human beings as well as all the animals, they do have the viruses which infect, but there are many which actually do not interact with R. They are the passive entity, and they just pass through us. Say, for an example, if the human feces is actually analyzed, the most common pathogens that are actually found are the viruses, but means that their numbers are the most because they are the smallest in size out of all the microbiological entities. And most of them are plant viruses. And there is a particular virus which is called the paper mottled virus. And these paper mottled virus essentially is one of the most abundant viruses that is essentially found. So this is basically uh, uh, we ingest those with the peppercorn. So essentially this is mottled virus that comes from the peppers that we usually consume, and this is the most abundant virus in the human fecal matter, and it is just an passive agent. It doesn't cause anything. But along with that, you can see that they are the vital members of the aquatic ecosystem, and they destroy many of the pathogenic organism as well in that way. They can be used to destroy the cancer cells. Many of the viruses are now being used to target drugs to the specific tissues or the cancer tissues. And there is an entirely different discipline which is known as the oncovirology. So this is oncolytic virology, where cancer, lytic means that which lies the cancer or kills the cancer. Oncolytic virology is entirely by a discipline on its own. And the bacteriophages in the human guts also may regulate many of the bacterial microbiome. And we call this association with the animals and the viruses as a virome. We always talked about the microbiome, which is mainly the bacteria. We understand that there are many different fungi that is also associated with that. But now we are trying to understand that how virus actually regulates, and the word virome is included in the microbiome as well. And they are important model organism as well, because many of these bacteriophages and the bacteria, they are easy to grow in culture, and many of these the infectious disease as well as many of the model diseases these can be used as tools for model organism. And the important part is that most of these molecular biological uh, discoveries were based on the specific process that the virus actually use. And there is an immense contribution of the virology into the basic understanding of eukaryotic as well as prokaryotic molecular biology. All right. So the viruses are they are absolutely acellular. So they do not have any kind of entity outside a living cell. And virology is the study of the viruses. So essentially, if we just talk about the viruses, these are infectious agents. Some of the, depending upon what they are, there are many viruses which doesn't infect anything. But these infectious virus is very unique because they are acellular. That means that they do not have a cellular entity, and they are all obligate intracellular parasite. Again, repeating, they are all obligate intracellular parasite. That means if they want to multiply, they need to be inside a living cell. Whether it's a bacterial cell, fungal cell, plant cell, or animal cell, doesn't matter. It needed to be inside the cell to replicate. Outside a living tissue or a living cell, a virus is just like a grain of sand. It doesn't have anything. So we do not use the anthropomorphic terms, which essentially we use for the living organism. So we do not use the word like dead or alive. We can just use the word rather more correctly that the viruses are inactive outside a living cell, or it is active inside the living cell. But sometimes, you know, that when we just talk about the virus, we use the word reproduce, multiply. These are anthropomorphic terms which we use for the living organism, and it is very difficult to avoid those terms. So just to make sure that the viruses do not reproduce, we are meaning that in presence of a cell, they multiply themselves by a process which we essentially call the reproduction. It is not reproduction by a living animal or anything like that. And essentially, the 2020s, the entire world learned about the impact of the viruses of the human health. And it is lately, but just imagine that 100 years ago in 1918, 50 million people were killed by the influenza pandemic. And then there were a couple of SARS that was a 2000 to 2003, about 800 people were killed by the original SARS. Then there was an outbreak of another disease, which is a MERS, which is a Middle Eastern Respiratory Syndrome around 2011-12, which is essentially spread from the dromedary or the Arabian camels and killed almost 2,000 people, and sporadically happening here and there, which is a very high lethal rate compared to the SARS-CoV-2 or the original SARS. So again, these are also used, as I said, that the model system in molecular biology as well. So what is the extracellular virus? They are inactive as such, cannot reproduce outside the living cell, where the intracellular virus essentially commandeered the host cells and they take over the cell. And this is totally by the chemical means. And we are going to understand that the viral nucleic acid take control and the proteins take control of the cell and synthesize the viral components, which essentially contain a nucleic acid and its other protein components, which mature progeny viruses are assembled and they are released by the host and will infect the neighboring cell and so on. So this is essentially what the viruses are. So the viruses can infect all cell types. Bacteriophages, simply sometimes called phages, infect the bacteria. Few archaeal viruses has been identified as well. Most eukaryotic viruses infect the plants, animals, even single cellular protists, as well as a fungi. So here you are looking at some of the viruses. They are very, very diverse, could be extremely small, but lately in the last 10 years, several of these giant viruses have been formed. So let's take a look at this. What we mean by the T-even coliphage. So the T-even means that their numbers are like this. They're T2, T4, T6, T8, and they are called the T-even coliphage. Why it is coli? Because they infect the E. coli, and this is a bacterial virus, which is the phage or the bacteriophages. These are extremely small, very, very tiny. On the other hand, there are several group of viruses. This is one of this is called this Mimi virus. There are several one of them, which is called a Mumu virus, Mimi virus, and these were all discovered by a French group. And then there is another one, which is called a Pandora virus. These are all the giant viruses. And these one of the giant viruses, like the Pandora viruses, is about 2 micron in length, and it is visible under the light microscope. So once upon a time, we used to use a definition that all the viruses can be only visualized under electron microscope. It is beyond the resolution of light microscope, but the giant viruses actually made the exception. This virus, particularly this Pandora virus, is visible under the light microscope. That is the only exception with these. And where we found these were actually drilled from the permafrost from the northern Siberia. When the scientists actually drilled the sample through the earth, these were actually frozen. And to whom they infect giant amoebas. So many of these viruses actually infect the protist or the amoebas. So these are the latest discoveries, and all of them were discovered in the last 10 years, 2011-12 onwards. So let's take a look at the virion structures, and we will define the word, what is a capsid, and we will define that what is an envelope. So the structure, essentially, when we talk about the virus, there are a couple of terms that we use. A virion is a mature virus particle. A virion is an infectious virus particle. Remember that. This is also important. Not only is it mature, but it is an infectious virus particle is known as a virion. And virions range in size. So essentially, this 20 nanometer in diameter is a very average shape. But remember that a virion of a virus can vary from, say, 20-25 nanometer to up to, say, a pox virus, up to a 200 nanometer or 300 nanometer in size. And the virions of course, the giant viruses can range up to 2000 nanometer, which is about 2 micron in size. So you can see that they could be very, very large as well. And the size of a bacterial cell is about 1.5 to 0.5 micrometer. You can see that some of the viruses are as big as those. And some of the viruses must be viewed with an electron microscope, most of the viruses, as we said. So first, take a look at that what the viruses essentially have. So what we are looking at, the first word that is important that we define is the capsid. So the capsid is the protein coat, mainly made up of protein, and sometimes it could be embedded lipid or glycoproteins, means proteins associated with carbohydrate. So this is essentially, think about that, this is the box of the virus. So think about that, this is the box, which is known as the capsid. Capso means the box. So from the box, inside the box, you have the nucleic acid. So the capsid essentially protects the nucleic acid, and we are going to see that the nucleic acid could be DNA or RNA. So the protein coat is a capsid. Now, there could be many different shapes and sizes. This is just a representative type. Sometimes these capsid could be embedded inside a membrane, and this membrane is of cellular origin. That means that this can be derived from some kind of endomembrane system in which the virus actually replicates. This could be the plasma membrane, this could be the Golgi membrane, endoplasmic reticulum membrane, and any membrane from the cell. Nuclear membrane, somehow, but this is a phospholipid bilayer endomembrane from a cell where it infects, it derives. And inside, when the capsid is enclosed inside this membranous structure, we call them as an enveloped virus. So this is a naked virus, non-enveloped or naked virus. This is essentially an enveloped virus. Whenever a virus is actually enveloped, the capsid is called a nucleocapsid. So the nucleocapsid here, when it is a naked virus, we call is just a capsid. Of course, it is a nucleocapsid, but we commonly call this as a capsid. But we specifically call the structure, which is a distinct entity of the structure associated or enclosing the nucleic acid, as the nucleocapsid, which is enclosed in that envelope. So very simple, that the coronavirus that you see all over the place is one of those which is the enveloped virus. So the nucleocapsid is inside. There's an RNA plus-stranded RNA genome inside that, and there is an endomembrane, and there are several different proteins. Out of those, the spike protein is a general term, but the spike protein is a trimeric protein, which is of the most interest. Along with that, there is a membrane protein and an envelope protein as well. But the spike proteins are the ones which attaches to the receptors and those. So if you just think about that, these spike proteins, okay, these are also sometimes called peplomers. They attach to the receptors. So how these how these viruses without these spikes can attach? Remember that these capsid proteins can be directly attaching to a corresponding receptor that during the entry of the viruses. So here we are looking at the size and the morphology of the different viruses. Here you can see that herpes virus, there are many different types, including the chickenpox virus, which is known as a varicella zoster virus or zoster herpes virus. That is actually a herpes virus. Along with that, there are many which causes this cold sores. Sometimes they called the warts. These are all caused by the herpes virus. On the other hand, this rhabdovirus is a bullet-shaped virus. The most common example of this rhabdovirus is essentially which comes from all these animals, or the rabid animals, is the rabies virus. So rabies virus is a classic example of the rhabdovirus. So here you can see that this is an enveloped virus with the nucleocapsid inside. This is also the nucleocapsid inside. This is an enveloped virus, whereas this T. even coliphage, this is a bacteriophage. This is a naked virus. Whereas here are you looking at a sulfophilus spindle-shaped virus. This is again a nucleocapsid inside, covered by an envelope, which is of course derived from some archaeal membranes. Human papillomavirus, and this is an HPV that you very well know, and this is also sometimes can be a cancer-causing virus as well. We have these HPV vaccines that are available and that can be sexually transmitted. And this human papillomavirus, or the HPV, is also a naked virus. Whereas here you can see that the mimiviruses, these are enveloped viruses with long proteins in here. This is a two-tailed virus. This is also a giant virus. And this is an acidina species as well. So they come in many size and shapes, as you can appreciate. So here we are looking at this as a more detail, that what is the capsid. This is a large macromolecular structure which serves as a protein coat of the virus, or basically the box of the virus, made up of protein subunit, and these are called the protomers. Many protomers can cause a capsid. So the smallest viruses, which encode very less number of proteins, and there are some viruses which only code just only one protein, or two proteins or three proteins. There, the capsid can be made up of just one kind of protein. Whereas some of the viruses actually code for 100, 200 different types of proteins, and some of them actually make the capsid. So the capsid is made up of multiple different types of proteins as well. So say, for an example, the adenovirus, which is a naked virus with spikes coming out of that, but they are made up of several different types of protein. They protect the viral genetic material and aid in transfer between the host cells. Non-enveloped viruses essentially construct a capsid. This is from many copies of the one protein, or in some of them, there are many copies of multiple protein. On the other hand, the enveloped viruses require the nucleocapsid protein and some of the extra protein to anchor to the membrane. And we actually do consider these capsids some specific type, having some specific type of symmetry. Now, for the naked viruses, we call them as an icosahedral symmetry. This is common. And particularly the naked viruses does have the helical symmetry. Does not mean helical means that they are coiled or something. They could be, but even the roundish structure which are enveloped are of helical symmetry. One of the classic example of the helical capsid of a naked virus is a plant virus, which was the first virus that was discovered, is a tobacco mosaic virus. So you can see that it looks like it is rod-shaped, but the symmetry is helical. So this is made up of these promoters, and they self-assemble into a rigid tube, and these protomers essentially influence the size of the capsid, and the genome, which is the RNA genome, is packaged inside these protomers. So this is a rod-shaped plant virus with the helical symmetry. Classic example is tobacco mosaic virus. The next one is essentially the icosahedral capsid. So the icosahedral capsid is essentially the structure of most of the naked viruses or non-enveloped viruses. So essentially what it is, an icosahedron is a classic structure which is a regular polyhedron. This polyhedron has 20 equilateral triangular faces. So essentially what happens, a typical icosahedral structure does have 20 equilateral triangular faces, and they have essentially 12 vertices. So that is a typical type of an icosahedron. So essentially, when we look at these icosahedron, the icosahedron essentially with this basic structure should have a typical type of symmetry, which is essentially a twofold symmetry. That means you can just separate these two equilateral triangles into 10 on one side, the 10 on the other side, by cutting them in half. You can have a threefold symmetry, essentially. So if you just look at essentially right here, from here, if you just look at a structure from here, you can see that there could be a threefold symmetry. Here you have a triangle, here you have a structure, here you have a structure, you have a threefold symmetry. And also there is a structure right here. If you just take a look at that carefully, you have one right here, 1, 2, 3, 4, and 5. So you can have a fivefold symmetry as well. So here is not very clear, but you can just easily imagine that there are these icosahedron, if you just draw it or properly, that they have a 2, 3, and 5-fold symmetries. On the other hand, so essentially, when a large particle is actually produced, now we have to do a little bit modification. So what happens that we are not going into the detail of the symmetry, but the simplest type of icosahedron, which is made up of these exact 20 equilateral triangles with 12 vertices, and they do have these 2, 3, and 5-fold symmetry. Now, there are some of the viruses which are more complex and larger and larger. When we essentially make larger and larger viruses, what we have that essentially along with this 2, 3, and 5-fold symmetry, we also have a 6-fold symmetry. So essentially, these are made up of either the pentamers or pentons, there are 5, or hexamer or the hexons, which is 6. Now, this usually causes a problem, technical problem, because what happens that when we are looking at these type of symmetries, say for an example, we are looking at 1, 2, 3, 4, 5. We are looking at 1, 2, 3, 4, and 5, and we have certain proteins in this space. I will essentially tell that what they are. So here you have 1, 2, 3, 4, 5. So here we have 1, 2, 3, 4, 5, and 6. So this is a 5-fold symmetry. This is a 6-fold symmetry. Now, let's take a look at what we can do here, that we can have a protein right here, which is right here. We can have another protein particle, which is right here. And we have another protein, which is right here. The other one was the bluish in color. So in the simplest virus, these proteins can be made up of by the same protomers. In some of the viruses, it could be the proteins which are similar, but they could be different proteins as well. But the problem is that what you are seeing, that when you are talking about these pentamers and the hexamers, you are fitting the same proteins right here in some places with a 5-fold symmetry, in some places with the 6-fold symmetry. So in this triangle, these proteins will essentially have a 6-fold symmetry, and in here, they will have a 5-fold symmetry. So what is the problem? The problem is that, think about that you have a tube which is in the ground, which does have these 5 sides. And think about that you have another tube which just snugly fits, which does have these 6-fold sides. And if you try to shove this into the 5-fold, it may fit, but the fitting is not exact. So what happens that the way that this problem of these pentamer hexamers of large capsomers are essentially solved, that these proteins, which essentially are the protomers right here, the protomers, they allow a little bit of flexibility of the arrangement of these proteins to accommodate the 5-fold symmetries and the 6-fold symmetries. So the proteins in the 5-fold symmetry interact with each other slightly different way compared to that how they interact with them in the 6-fold symmetry. And this is essentially what helps the building of a very, very large viral particle as well, with this flexibility of these, not only the 2, 3, and 5-fold symmetry, but also the 6-fold symmetry as well. So this is essentially an important consideration, that when we talked about the large viruses made up of these pentamers or pentons, or the hexamers or the hexans. So there is a word that is not needed for that, but sometimes what is called that a protein which is compared to with a 5-fold, when it is present in a 5-fold symmetry versus 6-fold symmetry, sometimes is used a word that how they are arranged in there, and we use a word which is called quasi-equivalence. We use quasi-equivalence. What we mean by that a protein so it is present in a 5 and is also present in a 6. So if it's the same protein, we say that the protein in the five-fold symmetry is quasi-equivalent to the protein in the six-fold symmetry as well. Means that they are not exactly the similar way interacting with its neighbors, but they are pretty similar. So this flexibility enhances, basically allows the building of large viral particle. On the other hand, what we look at, that viruses also do have these complex symmetry. And these viruses do not fit into the category of the helical or the icosahedral symmetry. You can see that this is a pox virus, largest of the animal virus, almost like 200 nanometer. And this interior is an ovoid brick-shaped structure. Large bacteriophages, which does have a binal symmetry, head resembles icosahedral, but the tail is actually helical. So this is called a binal symmetry. So you can easily see that they come in many sizes and shapes. So right here, this is an example of the complex symmetry where the head is icosahedral and the tail is helical, and then you have asymmetric structures, which are the tail fibers. That essentially what you see. On the other hand, when you are looking at these viral enveloped or the enveloped viruses always have the helical symmetry. They could be complex as well, but they always have the helical symmetry. Just you have to know that the viral viruses with the envelopes never have the icosahedral symmetry. But the enclosed nucleocapsid may have icosahedral symmetry or helical symmetry. So I'm repeating, the viruses which are the enveloped viruses, the enveloped viruses are always of the helical symmetry or sometimes could be complex symmetry, whereas the nucleocapsid, which is enclosed, could be icosahedral symmetry or it could be helical symmetry as well in some cases as well. So here we are talking about these, already talked about this, bound by the outer flexible membranous layer, which is called the envelope. This is the icosahedral capsid, you can see, and there is a protein called tegument protein. There could be some of these proteins present in between the capsid and the membrane is enveloped glycoproteins. Here you are seeing this essentially looks like a virus which is very similar to many different types of viruses. Of course, the coronaviruses looks like that, but influenza virus will also look like that. The important part is that you can see that out of the envelope, these are the spikes or the peplomers are sticking out. Many viruses actually do have this kind of symmetry. This is a helical symmetry. This is also a helical symmetry, but you can see right here, this is a icosahedral symmetry. This is not very clear. The inside nucleocapsid could be icosahedral or helical. And essentially, the animal virus envelopes, which are made up of lipids, which is essentially the phospholipids derived from the cells. And of course, there are many carbohydrates attached to these lipids as well as the proteins. So they are heavily glycosylated, and there are several proteins associated with this. So usually arise from the host plasma membrane or organelle membranes, as we said, which could be the endoplasmic reticulum, Golgi, as well as the nucleus, any membrane of endomembranous origin. So here the envelope proteins are virally encoded and may project the envelope on the surface, which are called the spikes or the envelopes, the spikes or the peplomers. And these spikes are involved in the viral attachment to the host cell, and they can have the enzymatic activity needed for entry to the exit from the host cells, and also sometimes used for identification of the different viruses depending upon what kind of protein they are. So after that, we are looking at the viral genomes are very structurally diverse, and we are going to talk about this much in detail in the later chapter. A virus may have a single double-stranded DNA or RNA. And when they have the RNA, we can have a double-stranded RNA, and the RNA could be of two different senses. It could have a plus sense, it could have minus sense. So why it is a plus or a minus sense that we have? The reason is that the plus sense RNAs can be directly translated sometimes by the ribosome. So they are translation-ready plus sense RNAs. Whereas the minus sense RNAs needed to be essentially converted into plus sense RNA, which you call as a mRNA synthesis, and then the mRNA becomes plus sense that can be translated by the ribosome. Where the DNA, on the other hand, doesn't have any problem. The DNA can be double-stranded, and the single-stranded DNAs could be either of a minus or a plus sense, but the plus and the minus sense DNA are essentially the same. You can just look at from different ends, but that is not the case of the RNAs. So the size of viral genome could be as small as 4 kb. Some of these calcivirus and parvoviruses have very, very small genome, and it could be even 2 megabase pair or 2 million nucleotides for some of the giant viruses as well. The pox viruses, herpes viruses also are very, very, very big, which also do have 200, 300 kilobase pair genomes as well. So the genomes can be, again, linear, circular, and some of the RNA viruses also do have segmented genomes, and we're going to talk about that in the next part. So next, we are going to talk about the viral life cycle. And the earlier, this experiment was done with the T4 bacteriophage in E. coli, and this was done in 1939 by Max Delbruck and then Emery Ellis. And what they did, that they essentially infected a culture of the E. coli with this bacteriophage and essentially took the time points right here of the sample. So essentially you have multiple of these samples which are infected at the same time, and right here they counted these phage. So there is a process by which you can count this phage, and we are going to see that in the later part of the chapter. So they took sample over the time. So essentially what they were found, that immediately after infecting the E. coli with the phage, there is a period where it looks like the virus actually disappeared from after the inoculation. There is no virus, and this is called an eclipse period. After that, what it was found, that it was shown that after some time, the intracellular viruses actually increase, and then they burst and come out of the cell. So why there are two graphs? What it essentially shows, that this is the viruses which is essentially inside the cell, whereas these are the viruses which are shown that is coming outside the cell as well. So essentially, why this eclipse period happened, and no new viruses present in the host cell. And this is the new viruses produced per infected cell when they are coming out. The first step and the evidence of the understanding of the viral life cycle is that this growth curve of the virus does not follow typically how a bacterial cell actually divides. So the bacterial cell will essentially have a lag phase, and then you have a log phase, then you will have a stationary phase and the declination phase. But the viruses do not follow that. They do have an eclipse period after you have a straight burst period, and we call this as a burst. So after the disappearance, this burst happens. So essentially, what is happening at this point of time? So think about that immediately after the infection, the virus is taking control of the cell, and it is synthesizing all the proteins and the capsomers and the protomers, which are the component of the capsid. Once everything is produced at this end of the eclipse period, the viruses are being assembled into new viral particle. At that time, you do not see that much of the extracellular virus. You see this period of time, see the latent period. In the latent period, the viruses are inside the cell. They are being assembled, but still you do not have anything outside the cell yet. At certain point of time, when there is enough amount of assembled virus, they will essentially come out by bursting the cell. So that's what it is. Eclipse period is essentially when you are not finding any viruses outside the E. coli or inside the E. coli. But the latent period extends beyond the eclipse period, where you find in the latent period, the later part of the latent period, which is beyond the eclipse period, you do find intracellular bacteriophages being assembled inside the E. coli, but still you have to wait a little bit until the rise period, where the E. coli actually burst and the viruses actually come out. So this is essentially what is called a burst, which is instant and happens very, very quickly. And this is known as a one-step growth curve of the viruses. Not all viruses actually shows this one-step growth curve, because there are some of the viruses which essentially bud out of the cell without immediately killing or lysing the cell, where it is much more complex and it doesn't show the classic one-step growth curve. So how the viral multiplication actually happens? So there are several steps for them, and it depends upon the viral structure and the genome, and it is very, very complicated, very, very diverse for many different viruses. But there is a general consensus that you have an attachment or adsorption, entry into the host, synthesis, assembly, and the virions are essentially released. So virus particle absorbed, then entry of the virus, and the viral protein nucleic acids are synthesized, and there is an assembly and essentially release of the progeny virions from the cell. So let's take a look at the attachment first. The viruses require the host cell in which they multiply, and the ligand on the virion attaches to a receptor on the host. So here we know that the SARS-CoV-2 spike protein is a viral ligand that attaches to the human receptor called the ACE2. Now remember that these receptors for the viruses are not there on the cells of the host to attach the viruses. All the receptors on the plasma membrane of the different host does have an entirely different function. So this is essentially an angiotensin converting enzyme too. It has an entirely different function in the renin-angiotensin pathway for essentially for maintaining blood pressure along with the kidney output. Entirely different function. So all the different receptors are essentially proteins on the surface of the plasma membrane on different cells, which are responsible for different purposes. But say, for an example, LDL receptor essentially take up the low density lipoprotein that we know, the LDL. Many of the viruses use this LDL receptor to enter that. So the virus actually evolved in such a way that they can exploit some of these proteins on the surface of the plasma membrane of the host cell to get entry into the cell. So that's basically the idea. And the receptor determines the host preference, which we call as a tropism. Viruses will bind to specific tissue receptors. What do we mean by this? That if we just essentially ingest the plant viruses do not attracted or get attached to any of the receptors found in the human beings, or the animal viruses will not infect the fungus, bacteria, or that. So you have to have the proper receptor and the corresponding protein into the virus so that they are very specific. So there are some of the viruses which can even infect some of the primates like a monkey. It can be a cat or dog, but it may not infect the human being. So even in between the mammals or the higher mammals, slight difference is actually do exist, which we essentially call the tropism, essentially. So the cells which are essentially does have the mechanism to attach to a virus and it can internalize and support the replication, we call them as a susceptible cell to that virus. And the tropism include that. So the attachment and adsorption essentially is the first step. So the viruses are essentially bouncing around on these cells. And this first step is totally up to electrostatics. So essentially, these are all charged particles and the surface of the cells are charged particles, and they are just adsorbed onto the surface of the cell completely by this process. This is completely adsorption. So that this is the first step. Once they attach onto the cell surface, they roll, they move around, and then in that process, rolling, moving, bumping, they finally found this receptor. Once they find the receptors, and if they have a corresponding receptor, then there is a very specific type of interaction, which is a protein-protein interaction. So the first adsorption is electrostatic, followed by a specific protein-protein interaction. Some of the times that these receptor engagement can cause a proteolytic cleavage of some of the part of these receptor proteins, which actually some of the part of these viral proteins. So it cleaves a part of the viral protein, and that actually helps in the fusion of the virus with the plasma membrane, or help in internalizing the virus, which is entry into the host. So essentially, after attachment, the virus genome or the entire nucleocapsid may enter the cytoplasm. And in some cases, only the genome enters, leaving the capsid attached to the outside of the cell. That means that the virus can essentially deliver just only the nucleic acid. Sometimes the entire part of the genome is delivered, but is still enclosed. And there are three methods that is used. The fusion of the viral envelope with the host cell plasma membrane. This happens in what is called a neutral pH, because outside the cell, the pH is in between 7.2. So this happens in a neutral pH. The viral envelope or the membrane can essentially fuse with this plasma membrane. The next is the process of the endocytosis, where a receptor binding essentially triggers the entire virus is taken into an endosome, and in the endosome, there is an acidic environment, and that acidic environment causes the virus to fuse with the endosomal membrane and deliver the nucleic acid. And in some cases, the entire virion can be released into the cytoplasm from the endosome, or in some cases, the nucleic acid is only released from the endosome and the nucleocapsid remains into the endosome. So let's take a look at these, that what happens. The first one is the entry of the enveloped virus by fusion with the plasma membrane, and this happens as a neutral pH. These are the receptors. You can see that the membrane engagement of the viral spike protein, that leads to a certain conformational changes. Sometimes these conformational changes are done by some of the proteolytic cleavage of these proteins. Say, for an example, for the spike protein of the SARS-CoV-2, the engagement of the ACE2 of the spike protein triggers a proteolytic cleavage, and there is a protease on the surface of the plasma membrane called TMPRSS2, TMPRSS2, TMPRSS2. That TMPRSS2 essentially cleaves a portion of the spike protein, and inside the spike protein, there is what is called a fusion peptide is buried. This fusion peptide is a part of the spike protein which is not exposed outside and only exposed after that proteolytic cleavage happens. And this fusion peptide falls onto the plasma membrane, and this fusion peptide is made up of all hydrophobic amino acid. So this fusion peptide essentially fuses the membrane envelope of the virus with the plasma membrane, and that fusion causes the virion to be released or the nucleocapsid to be released into the cytoplasm. And from there, the genome will come out and take control over the cell. Here you are seeing that entry of the enveloped virus by the endocytosis. Here what happens, that these are sometimes which is called these clathrin-coated vesicles. See that these XX right here, these are the clathrins, and these clathrin-coated pits are constantly being formed on the surface of the cells. These are, whether empty or not, they are constantly being formed and recycled. So when these receptors which bind to the virus are found in the clathrin-coated pit, these clathrin-coated pits essentially take the virus inside, and this is called an endosome. These endosomes do have on the surface of these endosomal membrane, which we called as a vacuolar ATPases, V ATPases. These ATPases pump protons or H plus ion from the cytoplasm into the inside of the endosome, and this high pH essentially cause some of these spike proteins conformational changes, very similar to this way. This conformational changes exposes a fusion peptide which was usually being buried, and that fusion peptide fuses this membrane along with the endosomal membrane and the nucleocapsid is delivered into the cytoplasm of the infected cell. On the other hand, we are looking at an entry of a non-enveloped virus as by the endocytosis. The same where here you have the capsid. This is a naked virus, and this is also can bind to the cognate receptors, and a receptor mediated endocytosis right here, receptor mediated endocytosis can take the virus inside. See, this could be clathrin coat mediated. It may not be clathrin mediated. There are some of the different types, and we are not going into that detail, which we call that there are some flask-shaped structures are formed, which we called as a caveoli. And these are some of the structure which are called caveolin-mediated endocytosis, where there is no involvement of clathrin, like these clathrin-coated vesicles. So all different kinds. But again, the non-enveloped viruses can be endocytosed by this method as well, where it is bound to a receptor found in the clathrin and it could be internalized. But the overview of that it just goes into the endosome. And once it goes inside the endosome, the same way the pH of the endosome is acidic, and it causes some of the conformational changes into the capsid protein, and it loosens up and it aids to release the nucleic acid through the endosomal membrane. And several of these proteins which are associated with the virion, with the capsid, actually aid in this transfer of this protein. And after this, the transfer of this nucleic acid material, so the nucleic acid, essentially, depending upon where they are, it can be destined to the nucleus or it can remain in the cytoplasm. So most of the DNA viruses, the nucleic acid essentially goes to the nucleus because it uses the DNA polymerases of the host cell to replicate themselves. On the other hand, most of the RNA viruses, with some exception, the nucleic acid remains in the cytoplasm because the enzymes for replicating the RNAs are essentially synthesized or being carried by the virus by itself, because there is no mechanism in the eukaryotic system or the prokaryotic system that can make from RNA to RNA. So remember this, we can make RNA from the DNA, but we cannot make RNA from the RNA. So to make RNA from the RNA, these are specialized enzymes always coded or carried by the virus itself. So they set up these factories in the cytoplasm, whereas most of the DNA viruses replicate inside the nucleus using the host DNA polymerases and the replication transcription machinery. A few of the viral nucleic acid, which are the RNA, associated with the polymerases, RNA polymerases of virus origin, also goes into the nucleus. Say, for an example, the influenza virus segmented RNAs, they are RNA genome, and along with the genome, there is an RNA-dependent RNA polymerase of the viral origin is attached to their RNA genome, and that goes into the nucleus. That's an exception. This much detail is not necessary. Just the overview of the entry of the viruses is good enough. So after that, we are looking at the stage, which is the synthesis, that differs the most between viruses, and the genome dictates these events. The double-stranded follow the typical synthesis inside the nucleus. As we said, the transcription translation is by the host, and the translation is always by the ribosome. The RNA viruses must carry in or synthesize the proteins necessary to complete the synthesis. And these are what we call as an RNA polymerases of viral origin. And what happens, that interestingly, that as these RNA viruses set up these viral factories for synthesizing these RNAs, there is a stage where one RNA template is making another RNA on top of that. So this is a stage which is called the double-stranded RNA stage. Now, in the prokaryotic, eukaryotic, or the living system, the double-stranded RNAs are not found. So the cells actually do have a very, very efficient machinery to detect these double-stranded RNAs, because they can be detected by a certain type of receptors called TLR, or the toll-like receptors, and they recognize that this is a product of a viral infection and immediately call the immune cells and the immune system for the antiviral response. So for that to essentially escape, to expose these double-stranded RNAs to the cellular machinery, or the receptor mechanism, or the detection mechanism, the many viruses actually set up a viral replication complexes, these enclosed machinery for the genome replication. And the classic example of this is seen in the coronaviruses, which the SARS-CoV-2 is one of them. So here what we are looking at, that they actually modify the endoplasmic reticular membranes, and they form these vesicles. And inside these vesicles, the synthesis of these RNAs take place, which is the viral nucleic acid. And then within that, from that, the mRNAs, which are required for making the viral proteins, come out into the cytoplasm, and they make all the proteins required for the capsomers and the protomers to make the capsids. And the genome, which is here, is essentially enwrapped into the protein, but the double-stranded RNAs are never get out of these inner membrane vesicles to be exposed into the detection system of the cell. So only the mRNA goes out. So the mRNA is a plus-stranded RNA, and the mRNA, the cells cannot easily distinguish which is a cellular mRNA or a viral mRNA, because they are almost identical. Although produce different proteins, but they do have the same way. They have a cap, they have a poly A tail, they do have the same structures. On the other hand, the genome RNA may be a little bit different, but they are immediately enwrapped with usually a protein, and which is a nucleocapsid, and that is essentially assembled inside the virus particle in certain process, many different processes. So again, that's one of the way that these viral replication and transcription complexes essentially are make up viral factories inside the cytoplasm to escape from the cellular detection system of the viral infection. So essentially, there are tightly regulated gene expression and protein synthesis, and these viral proteins are synthesized into the cytoplasm, and they essentially go to a certain place into the cytoplasm, or they can be associated with the endoplasmic reticulum, Golgi, or other structure, or sometimes we call the ECIG, which is the endoplasmic reticulum, ERCIG, endoplasmic reticulum Golgi intermediate complex. Say, for example, the coronaviruses are assembled into the ERGIC, which is the endoplasmic reticulum Golgi intermediate complexes. So the synthesis happens, and followed by the assemblies. So there are some of the late proteins. These are synthesized at the very last, and essentially that is the product of tightly regulated gene expression, which is that there are some of the early genes which is essentially responsible for synthesizing the genome, the mRNA, and the different components. And then finally, the proteins for the assembly are synthesized the last, and it follows by a complex process. Say, for example, this is an assembly which is of the bacteriophage, and we call this was a sequential assembly. So what it means that if this first step is not done, this later stage will not be done. So it is done step by process, and we call it a sequential assembly. And each of these steps are reversible. That means if the base plate is not properly synthesized, it can go back and it will try to assemble it back again. So unless it is correct, the next step will not followed. And that essentially is the quality control. And essentially, you can see the tube and the sheath is formed, where is the pro head is empty. The DNA is packaged into that, and the tube and the sheath with the collar is attached with that, and the long tail fiber proteins are essentially assembled at the very last. And this is the assembly process, and we call this as the sequential assembly. On the other hand, there are several types of mechanism which we call as a concerted assembly. In the concerted assembly, all the processes actually happen simultaneously. But essentially, what happens that the structures or the proteins do have the affinity for each other, and they self-assemble. And this is one of the beauty of these viral assembly, that when they find the proper structure or their proper partner, which are, should be next to each other, they essentially self-assemble. So after that, we are looking at the virion release mechanism, and there are two mechanisms. One is by the host lysis of the non-enveloped virus usually, whereas the enveloped viruses uses sometimes what is called as a budding. So here you are looking at E. coli cell with the bacteriophages. You can see that their tube sheath and the collar essentially, and these are the bacteriophages actually formed inside, and you can see that the cell is lysed. This is completely open, and the bacteriophages are coming out. And some of these virus-associated structures essentially forms these pyramid-like structures, and essentially from the cellular membrane, they can just go out. Some of the viruses essentially are essentially release the progeny virion by lysing the cell. So most of the non-enveloped viruses, with a very few exception, essentially lyse the cell just like this, whether it's a bacterial cell or an animal cell, lyse the cell and come out. Whereas the enveloped viruses essentially release by budding. So they don't kill the cells immediately and then they bud out of the cell, but essentially over the period of time, the cells will be eventually killed. So two different mechanisms. So here what we are looking at in a virion release of an enveloped virus. So here you can see the viral nucleocapsid is essentially the nucleic acid enwrapped with the protein. That's the nucleocapsid. And right here is some of the viral protein which essentially assembled near the plasma membrane. So what happens that these proteins are synthesized in a certain way through the secretory pathway and then carried by the cellular machinery and embedded onto the plasma membrane. So see, these are the hemagglutinin right here. These could be the type of spike protein that is found in many viruses, including the influenza viruses. And inside that, there is a matrix protein, which essentially enwraps the nucleocapsid. So here you can see that this protein-protein interaction brings the viral nucleocapsid to close to this matrix, and this movement happens by many different mechanisms. There are these microtubular highways made up of microtubules, and many of the proteins like the kinesins or the dyneins, they actually move these inside to the cell. So mainly when they are going towards the cellular periphery, the proteins which are essentially involved in this process are the kinesins, which essentially carry these motor proteins, carry these nucleocapsids, and when they are brought onto the near to this matrix protein, they essentially promote this budding process. And here you can see a part of the plasma membrane right here, or similar to that, that is essentially engulfed. So here you are looking at the neuraminidase. So this is basically not the exact structure, because this is the influenza virus does have segmented genome, which is not very clear. But neuraminidase is also another protein that is responsible, that is present on the surface, and the hemagglutinin. This is your H and N, and they are present on the surface of the membrane. And here you can see that it buds out, enclosing the nucleocapsid surrounded by this the matrix protein, and this is the free infectious virion with the envelope, and you have the hemagglutinin and the neuraminidase. So again, not needed as such. What happens, the neuraminidase essentially is cleave some of these glycoprotein bonds, because what happens that there are many glycoprotein interaction between these, and the neuraminidase essentially, in the absence of the neuraminidase, these virions cannot be released. So some of these bondings that happen in between the glycosylation of these glycoproteins sticks these viruses along with the plasma membrane. But when the neuraminidase is present, they digest these or basically disrupt the interactions with this hemagglutinin and the different glycoproteins and release that. So what I'm trying to say that this hemagglutinin does have the affinity for many of these glycoproteins. So see that right here you have these hemagglutinins. These tend to stick to the other proteins. So even after release, these glycoproteins or the hemagglutinins will try to get attached to these proteins. And the virus-free infectious viral particle cannot be released and attached to the plasma membrane even after budding. So this small amount of neuraminidase, they disrupt this hemagglutinin and the glycoprotein, which are still left on the surface of the plasma membrane, and allow the release of the viruses. So here also, virus-encoded proteins incorporated into the membrane, nucleocapsid simultaneous release, envelopes arise from the host membrane, but maybe Golgi, ER, and others are possible as well. And the virus may use also the host actin tails to propel through the host membrane between the adjacent cells. And we see this particularly for the pox viral release. So there is an actin tails which essentially pops the virus out of the cell, and a portion of the plasma membrane extrudes, and at the top of these actin tail, the virus actually rides and it's pushed to the next cell. So next we are looking at the different types of viral infection. The virulent phages are essentially the phages which infect the E. coli and essentially rupture them, and they have one reproductive choice. And the classic example are the T4 bacteriophages, and they multiply immediately and released by the host cells. Whereas the temperate phages, on the other hand, say for example is a lambda phages, they have two reproductive options. They reproduce lytically as the virulent phages, that means they will lyse the cell, or remain within the host cell without destroying it for some time, and this is called a lysogeny. So this is essentially a temperate phage and the host relationship where the phage DNA is integrated into the viral DNA, and we call this stage as a prophage, form of the virus that remains within its host by integrating its DNA into the viral DNA. And these bacteria, which are called the lysogenic bacteria, the infected bacteria, gets immunity to that virus and cannot be reinfected. And the prophage is replicated and inherited with the genomes of the progeny cells. And this is one of the process that we are going to discuss, that why this is probably a part of the survival, indirect survival for these lysogenic viruses. Now, what happens that there is sometimes a trigger, which we call as an induction. Say, for example, sometimes some of these lysogenic bacteria can be exposed to the ultraviolet, and at that point, these prophage, this DNA excises itself out and convert themselves into the virulent phages, making the viruses, and they burst out of the cell. So essentially, from the lysogenic virus, it becomes the lytic virus. So they can be of both the types. So why these lysogenic bacteria viruses actually develop? So essentially, there are two different processes. One is that if there are too many viruses in the environment and too less of a bacteria, then the problem is that there is not enough bacteria to get infected. In that time, what happens if the lysogenic virus essentially become temperate and do not kill the bacteria can divide, and then finally it can allow the virus to replicate once there is enough amount of bacteria. Also, another thing is that when there is a condition which is not favorable for the phage to replicate, in that condition, if a lysogenic bacteria essentially survive, basically the lysogenic bacteria can survive that environment, it actually helps the genome of the lysogenic phage to survive a condition which is essentially detrimental for the survival of the bacteriophages. So here we are looking at the lytic and the lysogenic cycle. This is the typical lytic cycle. So suppose this is an E. coli, the phage DNA is injected through the peptidoglycan layers, there are some of the enzymes which are present in these tailpin, which essentially bores a hole through the peptidoglycan and the plasma membrane, and the DNA is injected by squeezing these neck or the sheath. So once the genome is inside, this will take control of the cell, and it will degrade the bacterial genome and will make many copies of these, and it will make the mRNAs to make all the proteins that is required, and it will synthesize many new phages, and essentially when it is full, it will lyse the cells and essentially infect the new E. coli. On the other hand, what happens that sometimes in these phage, if it is a lysogenic phage, it can integrate right here like this, and right here, it can integrate the phage DNA into the genome of the bacteria, and we now call this as a prophage. And this prophage DNA is copied when the cell divide. But sometimes there is an induction process, such as an exposure to stress or UV, can trigger the excision of the host chromosome, and it can go into a lytic cycle like this. So the lysogenic and the lytic cycle can alternate as well. So when a temperate phage changes the phenotype of its host, this alteration is in the surface characteristic of the host sometimes, and the bacteria become immune to the infection. And we actually look at these salmonella when infected with this epsilon phage, they cannot be reinfected with the epsilon phage. So this is a kind of a survival strategy of a particular phage. To advantage, allow the viral nucleic acid to remain within a host, and when there is a high multiplicity of infection can occur, but the lysogeny allows the host survival in a few uninfected cells. This is what we talked about, that there are too many bacteriophage, but too less amount of the bacterial cell, which is called a high multiplicity of infection. Too many virus, too less cells. In that condition, the lysogeny allows the cell to survive and replicate to many individual bacteria, where the multiplicity of infections can go down to low, and then it can convert itself from lysogenic cycle to the lytic cycle for a productive infection. Archaeal viruses also can be virulent or temperate. Many can establish chronic infections in the archaea, and literally there is very little known about the mechanism to regulate their replicative cycle. We have started to understanding them essentially very, very lately. So on the other hand, when the infection happens to the eukaryotic cell, there are two different processes that can happen. What is called a cytocidal effect result in the cell death through the lysis. And also there are persistent on chronic infection. Many times after the infection, we essentially see a cytopathic effect, and the cells essentially die. They become star-shaped. They just crumble. And these are the degenerative changes or the abnormalities of the host cells. And here what we are looking at in an acute infection, essentially where the cells actually lyse the cells. And sometimes we do see a particular type of phenotype where multiple cells are fused into a giant cell. That is also a cytopathic effect. And these giant cells with multiple nuclei and large make up the viral factories. And these fusion of these multiple cells into large giant cells are also essentially formed. And these are essentially called syncytia formation. Syncytia formation. Syncytia singular, syncytia formation. These are essentially a giant cell formed by multiple nucleuses by the fusion of the cell, and the viral factories are essentially built up on those type of cells. Along with that, there could be also a latent infection where there is a slight replication of the viral genome or its component can happen, but the entire virus is essentially not synthesized. So this is essentially a stage which is there is no outcome of the disease essentially. So for an example, classic example, that if you have a chickenpox by the varicella zoster virus, the chickenpox virus or the varicella zoster virus essentially resides in some of our peripheral neurons of our peripheral nervous system. They are somewhere. and they are in a sleeping or the latent infection stage. Sometimes they can be activated, and we now know that due to stress and all those kind of things at a later age, and they can go into the active replication mode, and that gives you from chickenpox to shingles. So that is a classic example of the latent infection. On the other hand, there are some chronic infection where there is a slow release of the virus without the viral death, and some of the hepatic infections, we actually do see some of the viral hepatitis actually do have these slow release of the virus without the cell death as well. And also, there are several different viruses which are known to be oncogenic. That means that these viruses, there are two different viruses, and one group of viruses, essentially what they do, that they essentially turn on a proto-oncogene, essentially. So these proto-oncogene are essentially cell cycle regulators, and they turn on these proto-oncogene, so the cell cycle regulation goes completely uncontrolled, which leads to the transformation of the cancer. On the other hand, there are some of the viruses, what they do, that they turn off the tumor suppressor gene. So the tumor suppressor genes are the genes which essentially are always on and doesn't allow uncontrolled cell division or cell growth. So when the inactivation of the tumor suppressor gene or the tumor suppressor genes are turned off by some of the viruses, that can also lead in uncontrolled growth, and that can lead to transformation of the malignant cells and the cells. So some of the hepatitis C viruses can cause liver cancer. Human papillomavirus can cause also the cancer. So these are some of the oncogenic viruses. Some of the retroviruses can also cause cancer in many different animals as well. So these are some of the herpes viruses Kaposi sarcoma viruses can cause the cancers as well. So there are several different viruses which can be oncogenic viruses as well. So after that, we are looking at that how usually culture the viruses and how you enumerate them. One of the classic way that particularly they cannot be cultured like the cellular microorganism, and the lytic viruses are simpler as you can mix the virus and the bacteria in a broth. The temperate viruses require an additional step inducing the lytic phase, essentially. So you can just grow the E. coli and infect with the lytic viruses or the lysogenic viruses, and you can grow the bacteriophages fairly simply. On the other hand, you can enumerate these viruses also, and we call this as a plaque assay. And this is observed as the Petri plate is used to plate a lawn of the animal cells, or right here is an E. coli. So essentially, right here is an E. coli lawn. And after that, you have added a certain concentration of virus onto this plate. And you measure that amount of the viruses, and you do the serial dilutions experiments. So you have different dilution of the viruses, and you take a small amount of that particular dilution and you add to the plate. Now what you do, that essentially the idea is that one of these plaque means that right here, all the E. coli cells have been lysed. So you can count the number of plaque, and you can see that if you have a particular dilution of this plate, say for example, it is written somewhere in the plate, but again, if you just have a particular dilution of the virus, and you say that, okay, all right, I just got like 100 plaques out of this dilution, and you just replicate the experiment, so you can just easily tell that 100 plaque means that you have 100 individual viruses actually was added. So your 10 to the power, say 7 or 10 to the power 8 dilution does have the 100 viruses. So you can easily determine that 1 ml or 1 microliter of your viral sample will have how many viruses essentially. Essentially, you have to dilute these viruses to very high concentration, 10 to the power minus 9, 10, 11, 12 sometimes, and to find these, because if you add too many viruses, you are not going to see these individual plaques. It's going to destroy all the animal cells or the bacteria here in this example, in this process. So how this essentially happens, we are going to see in a second. First, we are taking a look at the cultivation of the viruses. So the animal viruses can be, the fertilized eggs can be incubated 6 to 8 days, and different parts of the egg can be injected by the viruses. And essentially, most of these uh, influenza viruses used to be essentially made into the fertilized chicken eggs in the industry. But lately, they are all of animal cell culture origin. And probably in a few years, they are going to be made in transgenic tobacco plants. So essentially of plant origin. So the molecular biology and the virology essentially can, over the period of time, have changed. And we find that now, if we just can make them in the transgenic tobacco plants, some of these viral proteins as the vaccines, they are probably going to be much, much cheaper. So we do not have any influenza vaccine anymore produced from the fertilized chicken eggs. They're all from the tissue culture or the cells. And of course, you may have these cell cultures in monolayers. Some of these animal cell culture are in the suspension and grown in what we call as a fermenters, 250, 500 liter large amount of fermenters, where you collect, and essentially you can lyse the cells and collect the viruses and concentrate them if you need the whole viruses. For some of the vaccine, you need the whole viruses, so you just have to collect the viruses and inactivate them and use them. For some of the other vaccines, it's a different process, which you need a protein or a combination of proteins. But here we are talking about the cultivation of the entire viruses in this process. And the cytopathic effects can be observed, where you know that at that point you have to collect the viruses. Plant tissue culture separates cells and the plants in the protoplast culture. So essentially you have to abrade a plant so that you make holes on their cell wall so that the viruses can enter. The viruses cannot penetrate the plants through the cell wall, so you need to abrade them so that the plasma membranes are exposed and the plant viruses can infect them. And rubbing mixture with an abrasive, and these cause localized necrotic lesion and generalized symptom of infection. So this is the plaque acid. We are coming to the picture of the plaque acid, that what we have seen here. So this can be done with the bacteriophages with E. coli, or this can be done with many other animal viruses. Not all viruses produce plaque either. So what you do, that you do the dilute the virus samples made and plate it with the appropriate host cells, and the number of plaques are counted. So essentially what you're going to see, that right here, these are the bacterial cell or the animal cell, and you have a particular phage or the animal virus. So this particular phage, if it is a case of a bacteria, is going to reproduce in this cell and is going to infect the neighboring cell. That is going to neighboring cell, that is going to kill. And essentially what you are seeing, that a single plaque is a round area where the cells, either the bacteria in this case or the animal cells, are killed. And those appear as a clear structure or the clear area compared to this background. So these results are essentially expressed as plaque forming units, or PFU. So you can, and they are directly proportional to the number of viruses. So this is just from one. So you can argue, if there are 2 or 3 viruses in here, probably the result is going to look like the same. That is true, but what happens that you usually do these experiments in many replicates, and you essentially statistically remove those kind of calculation mistakes that can happen in any experiments. So after that, we are going to talk about the viroids and the satellite. These are the nucleic acid-based subviral agents, and the viroids are essentially naked circle or structures of only RNA. These are closed circular single-stranded RNAs, which could be of the size of in between 250 and 430 base pairs of RNA. They do not encode any proteins, and their replication requires a host cell, and they cause some of the plant diseases. And they are essentially, the mechanism of the disease is probably that they do this RNA silencing. What it means, that suppose a gene is responsible for producing an mRNA to produce a certain protein for the plant function. These viroids can essentially silence in certain genes by binding to their mRNA, and that protein will not be expressed, and a pathogenic condition in the plant may arise out of those. So these are called the naked circles of RNA, and it is essentially believed that the viroids are the relics of the RNA world, essentially because the viruses or the or the DNA viruses actually arose after the RNA viruses, and these are the relics of the RNA world or the RNA viruses essentially. On the other hand, there is another type which is called the satellites. These are infectious nucleic acid, DNA or RNA, and they are enclosed in a capsid, but they need a helper virus to replicate. The satellite encodes its own capsid protein, but they cannot replicate on its own. They rely on a helper virus for the replication enzymes. And most of the satellite viruses use plant viruses as their helper viruses, and they enhances some of these diseases of the plant. But animal satellites have also been reported. Say, for example, there is a virus which is called a dependoparvovirus. This dependoparvovirus, so you can see that from the name, it depends on something. Dependoparvovirus is an animal satellite, and it uses animal viruses like adenoviruses, herpes viruses, or vaccinia viruses. So when adenovirus infection or a herpes virus infection or a vaccinia virus infection, this is the pox virus, happens, at that time, the satellite dependoparvovirus may essentially you can see the presence of these dependoparvoviruses. Sometimes it is seen that the disease enhancement or the severity of the disease are enhanced by some of these satellites. So that's pretty much about the viroid satellites and the viruses. The last part that we are talking about is the prions, which is another cellular agent, and it only forms from a protein. This is an infectious protein. And we actually discovered or known in the 1980s, where Stanley Prusiner actually discovered this prion protein, which is an infectious protein and can give rise to many different diseases like mad cow disease and all those kind of things. So what exactly it is? This prion protein is a normal protein, and you can see that this is a typical structure of a protein with most of it's in alpha helical form, where the green part is the alpha helical form, very little in the sheet or the beta sheet form. And this is a normal protein that is found in every cell, and this is a normal protein of our own. By some unknown mechanism, sometimes what happens, this protein may misfold. And when it misfolds, you can see that this protein forms an extensive beta structure. These are the beta sheets. So essentially, these extensive beta sheets, we call this disease prion protein, that they form these amyloid structures. And essentially, this is the disease prion, and this is infectious. This can turn a normal prion into a disease prion. And essentially, it embeds itself onto the plasma membrane of the neurons and essentially kill them. So how this is essentially developed? So these prion protein, which is known as a PRPC, prion protein cellular, present in normal form. The abnormal form is known as a prion protein, PRPSC. SC is a scrapie, which is essentially a neurological disease of the sheep known as a scrapie, and that protein of the disease protein is the PRP, prion protein scrapie, which is essentially this form. So this is PRPC cellular, this is PRPSC, which is prion protein scrapie. Only different in the 3D conformation. And the PRPC irreversibly converts to PRPC, and still unknown if the loss of this PRPC or the accumulation of PRPC lead to the disease, probably both. But we do have some models for that. And that's the reason that as we do not know exactly the mechanism, this is essentially a model. So first is a heterodimer model of the prion propagation. So here you have an endogenous PRPC. These are normal protein. If it interacts with a prion protein SC, the prion protein cellular converts into the disease form. And this accumulation of these prion protein SC can cause this neurological disease or the death of this neuronal cell. On the other hand, the other process is known as the fibril model of the prion propagation, which is essentially the still unknown loss of PRPC or accumulation of this leads to disease. In this process, the prion proteins essentially is converted into the prion protein SC, and this is an amyloid protein where they form these extensive amyloid structure or a fibril-like structure. And from these fibril-like structures, some of these amyloid PRPSC breaks and this cycle can go on. But if one of these prion protein SC essentially mixes with a prion protein cellular, it can convert it to an amyloid structure. So essentially from a fibrillar structure, it can break into smaller pieces to the new seed. So essentially that is. So these are both are the hybrid models. None of them actually essentially contradict each other, but this is essentially formed. So how it looks like on this level of the plasma membrane. So here you are looking at the plasma membrane. You can see that two leaflets. This is the outer leaflet of the phospholipid, inner leaflet of the phospholipid. And here is the prion protein cellular. And see that these are all the different modification or the glycoconjugation with the protein. And right here, this is the extensive alpha helical structure. And this is associated with a GPI anchor. This is a glycophosphatidylinositol anchor to the membrane, and a prion protein SC is a part of it. Now what happens that in this amyloid form, which essentially form these fibrils and accumulation, you can see that this is a GPI anchored protein, and numerous number of these prion protein SC fibrillar form, which is the extensive beta sheet, actually accumulated one over other, which is essentially this structure. And this association along with the GPI anchor completely distorts the membrane, breaks the membrane apart, and essentially this prion fibril kills the neuronal cell. And the neurons are amidotic. They do not have a replicating ability. And if a neuron is essentially killed, that portion of the brain becomes a void. It is a vacuole, and essentially in the progression of the disease, it essentially causes a spongiform brain that we are going to see. So here we are looking at a neuron with the healthy prion on the surface of it, monomeric high alpha helix. This is the prion disease, multimeric amyloid furvil, parallel in register architecture, high beta sheets, and these essentially form these plaques which essentially disrupt the plasma membrane, as we said, and essentially kill the neurons. And here you are looking at a cross-section of a neuron, all the white areas. So where the neurons are, these are the biggest cells. These are the, right here is a neuron, right here is a neuron, here is a neuron, here is a neuron. These are the neuron. And the rest of the cells are the supporting cells, which are called the glial cells of the nervous system. But where you are seeing these large white vacuolar structure, white spaces or the vacuoles, these empty spaces where essentially, so here is probably a dying neuron. These empty spaces were essentially occupied earlier by some of the living neurons. And this gives the brain a spongiform degeneration. And we call this that one essentially, one of the disease is called the BSA, or the bovine spongiform encephalopathy. And this is the pathology of the disease. So the prions can affect different parts of the brain. It is the same disease, but when it affects different part of the brain, it can essentially cause different disease. Variety of neurodegenerative disease in the human animals. It's called the scrapie in sheep, bovine spongiform encephalopathy in the mad cow disease. And there are several human diseases. So essentially here, thalamus, fatal, if the prion proteins essentially accumulate here and kill this area, the neurons, fatal familial insomnia. Right here is the classic disease, which is known as the classic the Creutzfeldt-Jakob disease. Whereas you can see that when it affects the brainstem, it can cause scrapie, bovine spongiform encephalopathy, and the chronic wasting disease. Kuru, when the cerebellum essentially is affected. It's called Kuru, which is a loss of this coordination. There's also another disease called the Gerstmann-Straussler-Scheinker syndrome that is also one of the neurodegenerative disease. Very interestingly, of the Kuru disease is that this was first discovered in the late 1950s, 60s in the Papua New Guinea. And in the Papua New Guinea, there is an eastern part of the Papua New Guinea. There is one of the areas where there are some of these tribes live in there. These are called the four tribes or the four people, Fore. And these are some of the highland areas. And what happens that there were some of these four tribes, they were having Kuru. And they had a ritual that usually if a person dies, they were cannibals. So they will essentially cut them up and then, of course, consume the human flesh. So unfortunately, what happened that there were many of these deaths that happened to Kuru. And when they ate this contaminated meat, there was almost an epidemic of Kuru in between 1957 and 1960. So documented, there were several documented, a large number of these four people actually succumbed to the Kuru disease because all of these prion diseases are completely lethal. Once it happens, there is no cure for this disease. So this happened, and we know much more later. We knew the disease, the neurodegenerative disease of Kuru from the 1960s, but we did not know what it caused or the causal of Kuru only after the discovery of the prion proteins, later part in the 1980s. We actually know those Kuru deaths were essentially happened by these prion proteins. And these prion proteins, particularly in this parallel high beta sheet form, they are almost indestructible. They cannot be destroyed even by autoclaving. You have to autoclave for more than 3 hours in presence of sodium hydroxide. No one cooks meat with that. So cooking or anything will not destroy those. So once a person, animal, or anyone gets a prion protein in a disease form, it is just unfortunately that person at one point of time will die from that disease. There is no cure. About 150 people so far actually died of this disease. There is no chance of that we can develop a drug against that. At least in recent times, there is no such research is going on, which actually even shed any kind of light that whether there is a possibility that somehow we can fight this disease. So this is one of the deadliest disease known to human being, just caused by a infectious protein particle, which is just folded or misfolded. That's about it. So essentially, this is essentially the complement to the chapters of the three domains of life, the cellular agents, how they infect or affect all the three domains of the life. And this is outside the three domains, but are very important to the microbial world, the virology as well as prion biology. These are integral part of microbiology. And we have seen that each other, the cellular entities, how they function, replicate, and can round out the greater understanding of the microbiology. And as I said, the virome is a new area of research. And of course, in terms of disease, after these pandemics and many of these debilitating viral disease, the virology is actually essentially a very important subject for years to come.